Hello everyone, my name is Janie Brandsma and this is my fourth and final project of the Spring 2023 semester for BIOLFY5099. I have chosen to talk about the sexually transmitted disease gonorrhea. For a brief overview on the etiology of gonorrhea, it is a bacterial infection of Neisseria gonorrhoeae that is passed from person to person during sexual relations or even during childbirth. Gonorrhea commonly infects the mucosal membranes of the lower urogenital tract, so the urethra, the vagina, the uterus, and the cervix, but it can also infect other areas with mucosa such as the eyes, throat, and rectum as shown in the images to the right. Gonococcal conjunctivitis is typically associated with neonatal gonorrhea, so with children, which is why it's important for women to be tested for STDs prior to childbirth. However, getting screened for STDs is important for anyone who is sexually active to prevent the spread of these STDs. Gonorrhea is the second most common STD in the United States, just after chlamydia. The incidence of this disease is approximately 1,000 people per every 100,000. And as you can see in the GIF on the top left, cases have rapidly increased from 2011 to 2020, likely because strains of Neisseria gonorrhoeae are developing antibiotic resistance. The age groups that are most affected by this disease are people around the ages of 15 through 24, most likely due to the lack of knowledge on the importance of wearing protection during sexual intercourse. In the image on the top right, you can see that statistically women acquire gonorrhea at a higher level than men. This is because there is a higher chance of transmission of this disease when an infected man passes it on to a woman. This is due to the increased amount of mucosa found in the female urogenital system. Symptoms of gonorrhea will show up anywhere from 1 day to 2 weeks after inoculation, but for many there are no apparent symptoms. Often, women will be misdiagnosed with a bladder infection or with a vaginal infection such as bacterial vaginosis. With the quantity of asymptomatic or misdiagnosed cases, it can lead one to believe that the incident rate is much higher than what is reported. Some symptoms for both men and women include penile discharge or excess vaginal discharge that is white, yellow, or green in color. Pain when urinating or pain during sexual intercourse are another symptom found in both men and women. In women, there can be abnormal bleeding in between periods, and in men, if the infection spreads up the urethra, causing epididymitis, testicular pain and swelling will occur. No matter if you have severe symptoms or if you are asymptomatic, serious complications can occur, such as pelvic inflammatory disease and infertility. To diagnose gonorrhea, samples must first be taken. For men, a urine sample will be taken, and for women, urine, vaginal, or endocervical samples are taken. From there, the samples can be cultured on either non-selective chocolate agar or selective Thyre-Martin media. Nucleic acid amplification testing is another method of diagnosing gonorrhea. Often, NAAT is chosen because of its efficiency and specificity to bacterial DNA. NAAT is done by performing reverse polymerase chain reaction on the sample with a primer that is specific to gonococcal DNA. From there, DNA from Neisseria gonorrhoeae will be exponentially copied, allowing for analysis and comparison to other gonococcal DNA. As I briefly mentioned before, gonorrhea is caused by a bacterial infection of Neisseria gonorrhoeae. When stained, Neisseria gonorrhoeae is seen as a gram-negative diplococcus. The sides of the gonococcus are compressed, causing the bacteria to look a little bit like a coffee bean. You can see this shown in the top right image of Neisseria gonorrhoeae inside 2 neutrophils. Neisseria gonorrhoeae is a facultative aerobe, meaning it can grow in either oxygen or without oxygen. It does have a preference for microaerobic environments, or environments with small amounts of oxygen, such as in the cervix or the urethra. A distinguishing trait of Neisseria gonorrhoeae is that it exhibits many pili, as shown in the image in the top left. These pili have an important factor in the virulence of gonorrhea and its ability to survive in the human body. Gonorrhea is best cultured in either the non-selective chocolate agar or the selective Thyre-Martin agar. Thiamartin agar is easily able to isolate Neisseria gonorrhoeae because it is infused with antibiotics that inhibit the growth of other bacteria found in a patient's sample. Gonorrhea is a multi-drug resistant bacteria which allows it to grow on Thiamartin agar, unlike many other microbes. Gonorrhea is able to colonize mucosal membranes, which are usually found at entrances to the inside of the body. These areas include the intestines, throat, and respiratory tract, but importantly at the eyes, uterus, cervix, penis, and urethra. The mucosa is formed of either simple columnar epithelium or stratified squamous epithelium, depending on which mucosa is being viewed. You can see an image of these types of epithelium above. The mucous membranes function to lubricate these surfaces to prevent abrasion from particulates or from friction caused by other surfaces. The mucus produced by glands from the mucous membranes allow them to have this lubricating effect. It also functions as a part of the immune system by protecting the body from pathogens that may enter the body. Mucosal membranes often have a high level of immune cells such as neutrophils and macrophages because they are at the interface between the interior and the exterior of the body. In the female reproductive system, the endometria is a very important mucosa. It functions to protect developing eggs or to remove unfertilized eggs. When the endometrium sheds, this is what causes menstruation. Now I will talk a little bit more about the immune system. The immune system works to defend the body from pathogens such as parasites, bacteria, and cancerous cells. The innate immune system is the first line of defense for the body, giving a fast general response to any pathogen. From there, the adaptive immune system will kick in with a more specific response to a pathogen. The adaptive immune system contains cells such as memory B cells and T cells that are able to remember a specific pathogen, allowing for an efficient response in the future. There are many immune cells, but I wanted to focus on the immune cells that are affected by gonorrhea. Macrophages are an important cell that are found in both the innate and adaptive immune response. Macrophage is able to phagocytose anything that is unwanted in the body, like pathogens or dead cells. It also functions as an antigen-presenting cell, displaying an antigen to a lymphocyte, such as a B cell or a T cell. This is what turns on the adaptive immune response. Macrophage is able to trigger an inflammatory response by releasing the cytokines interleukin 1, 6, 8, and 12, and importantly, tumor necrosis factor alpha. Neutrophils have a similar function to macrophages in the innate immune system in that they are also able to phagocytose antigens. A difference is that neutrophils are granulocytes, meaning that they contain granules that act as enzymes, allowing for the destruction of pathogens. Plasma cells, or B cells, are an important white blood cell in the adaptive immune system. They are able to produce specific antibodies in response to an antigen in order to neutralize or flag them for destruction. Neisseria gonorrhoeae is able to infect the epithelium of mucosal membranes, and there are many virulence factors found on the outer membrane allowing it to do so. With the use of its many type 4 pili and opacity-associated proteins, it is able to adhere to the epithelial cells, allowing for colonization. Neisseria gonorrhoeae is a master at evading the body's innate and adaptive immune systems. Neisseria gonorrhoeae also uses its pili to prevent phagocytosis from macrophages and neutrophils, allowing for the bacteria to continue to reproduce and grow in the mucosa. Unique to gram-negative cells, Neisseria gonorrhoeae contains lipooligosaccharides, or LOS, similar to lipopolysaccharides, which are found on the outer membrane. The lipooligosaccharide is able to stimulate macrophages, neutrophils, and other immune cells to secrete a large volume of tumor necrosis factor alpha, which can be harmful for healthy cells. Neisseria gonorrhoeae is able to produce the enzyme immunoglobulin A protease, which binds to immunoglobulin A, which renders it useless in the adaptive immune response. The type 4 pili found on Neisseria gonorrhoeae is a very important feature of this disease. The pili are able to adhere to epithelial cells, allowing for the bacterium to be stably attached during reproduction. After binary fission, the bacteria begin to clump together, allowing for the formation of a biofilm across a mucosal surface, which allows the colonies to be able to overcome stressful environments. Another function of the pili is that it allows movement across a mucosal surface. The type of movement done by Neisseria gonorrhoeae is called twitching motility. It is done by a series of elongation and retraction of the pilus, allowing the bacteria to be pulled forward. You can see a cartoon version of this being done above. Twitching motility allows for the bacteria to spread further up the urethra or into the fallopian tubes. A very useful function of these gonococcal pili is that they are able to perform horizontal gene transfer. When a pilus connects with another microbe, it is able to take a gene, allowing for Neisseria gonorrhoeae to be better adapted to its environment. Horizontal gene transfer is an important mechanism in the spread of antibiotic resistance among bacteria. The structure of the type 4 pili is a polymer formed by thousands of pilin protein subunits, which you can see on the left image. Each pilin subunit is composed of 165 amino acids. The subunits bind, forming a helical formation with the hydrophobic amino acids at the core. On the right image, you can see a ribbon model and a space-filling model of a gonococcal pilus and how each subunit, shown in a different color, can form a spiral shape. A single pilus has an average length of 0.9 to 1.2 micrometers, but it has been found to be as long as 10 micrometers. At residue 68, there's a phosphoserine, which is theorized as to why gonorrhea is able to adhere to epithelial cells. Opacity-associated proteins, or OPA proteins, are what cause Neisseria gonorrhoeae to form opaque, grayish colonies, hence the name. You can see an image of Neisseria gonorrhoeae on chocolate agar in the top image to the right. OPA proteins allow Neisseria gonorrhoeae to survive and adhere to mucosal membranes. It is able to adhere to epithelial cells and neutrophils by binding to carcinoembryonic antigen-related cellular adhesion molecules, or CCAMs, which act as a receptor between host cell and bacteria of the healthy mucosal microbiome. This tricks neutrophils into believing that they are a part of the normal flora in this environment, allowing for the bacteria to both adhere to cells and avoid phagocytosis. OPA proteins are able to turn on and off the gene that encode for the OPA proteins in a process called phase variable expression. Similar to the pili, the OPA proteins are formed by several subunits. Each subunit of the OPA protein is approximately 238 amino acids long. You can see the ribbon model of an OPA protein subunit in the top image to the right. On Neisseria gonorrhoeae, there are 7 to 9 unique OPA proteins. The types of proteins differ in the amount of extracellular loops, which allows for them to bind to different CCAM receptors. All of the OPA proteins share a beta-barrel structure that forms the transmembrane segment, allowing for the OPA protein to stay firmly bound to the gonococcal outer membrane. On the bottom image to the right, you can see an image of the crystal structure of a complete OPA protein. The lipo-oligosaccharide, or LOS, found on the outer membrane is unique to only gram-negative bacteria. The LOS is able to induce an inflammatory response by activating the alternative complement pathway of epithelial cells. The activation of this complement pathway triggers macrophage to start secreting the cytokine tumor necrosis factor alpha. However, Neisseria gonorrhoeae is able to disguise itself by sialation of the LOS. Sialic acid is produced by the host epithelium, and with the use of the enzyme sialtransferase produced by Neisseria gonorrhoeae, sialic acid will cap the end of the LOS, enabling it to be camouflaged from macrophages and the complement pathway. The high amount of tumor necrosis factor alpha produced will no longer be effective on the bacteria and will then cause host cell damage or apoptosis. Before transmission from a woman to a man, the gonococcus will become desialated in order to infect the urethra. The LOS is composed of 3 oligosaccharide chains, alpha, beta, and gamma. Unlike the lipopolysaccharide found on many other gram-negative bacteria, the LOS is missing the repeating O polysaccharide side chains. The LOS is firmly attached to the outer membrane by the transmembrane lipid A that you can see on the right of the image that I have included. Lipid A is connected to the oligosaccharide chains by the 3-deoxysugar monosaccharide KDO, which is then connected to heptose 1. Silation occurs at the alpha terminal galactose residue by a 2,6 or a 2,3 linkage. Immunoglobulin A1 is a dominant antibody produced by plasma cells found at mucosal membranes. Immunoglobulin A protease acts as an enzyme that hydrolyzes the peptide bonds of the heavy chain of immunoglobulin A at the hinge regions. I have pointed to this in the image on the right. We know that this is the case because in patient samples of gonorrhea, the parts of the split antibody were found. Because the antibody has been split, it is no longer able to flag gonorrhea for destruction by immune cells. Immunoglobulin A protease is a large enzymatic protein that is produced by most all strains of Neisseria gonorrhoeae. It is composed of 1,958 amino acids. On the left image, you can see the structure of an unbound immunoglobulin A protease. This enzyme is formed by several alpha helices, but near the active site there is a large beta sheet, which I have pointed to in the left image. On the right image, you can see the immunoglobulin A protease in teal interacting with the immunoglobulin A antibody in pink and green. Over the years, Neisseria gonorrhoeae has developed antibiotic resistance from all major classes of antibiotics except for one antibiotic from the cephalosporin class. This includes resistance to all penicillins, sulfonamides, tetracyclines, quinolones, and macrolides. The reason why gonorrhea has quickly developed antibiotic resistance is because it is able to perform horizontal gene transfer with the use of its pili. Nowadays, antibiotics are used very frequently or used incorrectly by not taking the complete amount prescribed, which allows for bacteria to develop immunity to the antibiotic. The pilus of Neisseria gonorrhoeae will interact with another microbe, allowing it to take the gene that encodes for the resistance of that antibiotic. The genome of Neisseria gonorrhoeae is very adaptable and will frequently and spontaneously develop mutations. Some mutations are able to give rise to new antibiotic resistance. The image I have included is small, but it shows the history of antibiotic resistance in gonorrhea. Currently, the only effective treatment for gonorrhea is a 500 mg dose of ceftriaxone injected intramuscularly. However, in 2011, the first ceftriaxone-resistant strain was discovered in Japan. When treating gonorrhea, the antibiotic used must be beta-lactamase resistant to prevent degradation by the beta-lactamase produced by Neisseria gonorrhoeae. There has yet to be a vaccine developed for the prevention of gonococcal infection, but scientists are working on one with the use of the lipo-oligosaccharide produced. It is very possible for someone to reacquire gonorrhea because the immune system is not able to build an immunity against it due to the fact that it is able to hide from immune cells. There is fear that in the future, gonorrhea will become a superbug that cannot be treated. While there may not be very many treatments for gonorrhea, there are preventative measures that can be taken, including wearing a condom or dental dam during all sexual relations or abstaining from sex as a whole. To summarize what you have learned during this presentation, I would like for you to match each of the virulence factors listed on the left with the correct functions or function listed as 1 through 6. I would also like for you to match the correct image of the structure listed as A through D. Some of the functions will be used for multiple of the virulence factors. Here are the correct answers so you can check your work. And these are all of my references that I used while making this protection. Thank you for listening, and be sure to use protection.